The mechanism of muscle contraction is explained by the sliding filament theory. This theory states that muscles contract when actin myofilaments slide over myosin myofilaments, shortening the sarcomere.
Whoa, Simon! How are you able to lift such a heavy object?
It's easy, it's muscle contraction.
A muscle contraction?
Yeah, you see what happens is a motor neuron carries an action potential from the brain until it reaches a muscle tissue.
A neuromuscular junction is where a presynaptic terminal of a motor neuron meets a postsynaptic membrane of a muscle fiber, also known as a sarcolemma.
An action potential causes a neurotransmitter called acetylcholine to be released into the gap between the neuron and the sarcolemma.
So the acetylcholine is like the key to the door for salt?
Yeah. This allows the action potential to be transferred from the neuron to the muscle cell. Now, the muscle action potential travels along the sarcolemma and through the T tubules. There are cavities that allow impulses from the membrane to penetrate the cell and activate the release of calcium ions from the sarcoplasmic reticulum.
The sarcoplasmic reticulum?
Yeah. The calcium ions then flood into the cytoplasm of the muscle cell, also known as the sarcoplasm. Calcium ions bind to troponin, which causes it to change shape. This then causes tropomyosin on the active filament to move to a different position and expose the myosin binding sites. The myosin head binds to the myosin binding site, releasing ADP and phosphate. The myosin-actin crossbridge rotates towards the center of the sarcomere. This is known as working stroke, which causes muscle contraction. ATP binds to the myosin head, which results in the myosin head and myosin binding site to detach from each other. The myosin head acts as ATPase, which breaks down ATP into ADP and an inorganic phosphate so that the process can be repeated. If there are no further action potentials, then the number of calcium ions will decrease. This then causes troponin-tropomyosin complex to move to its original position, blocking the myosin binding sites, which then causes the muscle to relax.